for those of us that want to accelerate photosynthesis or optimize photosynthesis, it's very, very simple. That if we decrease stress, photosynthesis is going to come on its own. Plants are made to grow again, and they're made to grow on well, we'll learn about that next week, but they're made to grow typically in symbiotic relationship with microbiology with carbon, right? In the root zone. It's cycling carbon through the plant, through this back to the soil, through the microbes and back. This, if you look into some of the research that, that maybe is in Acres magazine or some of the other academic literature out there, it consistently shows that these symbiotic relationships between plants and microbes are key to decreasing plant stress and buffering the capabilities of the plant to uptake nutrients. When photosynthesis is slowed, that decreases your yield, right? It's pretty straightforward. When your plants can't actually physically utilize the sun that's beating down on them, you're limiting your yield. And so, there's a lot of people out there that say, oh, well, leaves are solar collectors, right? And that's very, very, very true. But if those leaves aren't on, or if there's dust on them, or if there's some, cells in, photocells that aren't actually performing properly because they're stressed out or they don't have the adequate water circulation or so and so forth, that's going to decrease your yield. And it also is not going to just decrease your yield, but it's going to decrease quality, survivability, and increase plant susceptibility to further stress. So that's the other thing. So stress begets stress, right? And health begets health. And so the more that we really drill down on how do we alleviate that stress, instead of saying, well, what does that plant actually want, guess at it for the 4 R's or whatever we're trying to do. If we incorporate a component of how do we alleviate stress through fertilization, now all of a sudden the game changes. And that balanced nutrition that then comes in those sort of a feeding program that you're able to develop from leaf analysis then will help to limit the longer term susceptibility to undesirable characteristics on your farm. So we have been very vocal speaking out about plant nutrient excesses, and it's a very key part to our agronomy and has really helped us to achieve some significant results for a lot of our growers. But, how do you find those? How do you find where the excesses or the deficiencies are located within a plant at a given moment, right? And so ultimately it's, if you go to any of the leaf reports, when the old leaves are lower part per million value on than the new leaves of your macronutrients, those are going to be deficiencies. Conversely, the old leaves are have a larger value than in parts per million than the new leaves of excesses such as sodium, chloride, or aluminum. So in other words, if you have, we'll call it 3 parts per million of aluminum on the top in your new leaf, and you've got 45 part per million of aluminum on the bottom of the plant, that's a significant excess, right? Versus if you had, we'll call it 25 part per million nitrate in the new leaf, and you had 2 part per million nitrate in the old leaf, that would be a deficiency of nitrate. So it gets, it gets pretty deep pretty quickly when you start to look at new leaf deficiencies, old leaf deficiencies, new leaf excesses, old leaf excesses. But there's actually 4 things happening in every SAP test, and identifying that each one of those step by step, and then developing fertilization programs from those is very, very key, or can be very key to crop success and ability to steer a crop during season. So if you get into the micronutrients, and a lot of studies and infographics out there kind of talk about this, that when the new leaf part per million value of manganese, boron, iron, zinc, cobalt, or copper is higher than the old leaf, you've got a excess of those micronutrients. Similarly, where you have the new leaves of a micronutrient lower than the old leaves in part per million value, that indicates a deficiency. So for instance, say take something like copper, where if you want to identify, well, have I sprayed too much copper on my plants because I was using it for a fungicide? Well, if you have a copper, 75 in the new leaf and the old leaf has 3 or 5 or 7 or something like that, then that's typically a short-term acute copper stress. The plant will metabolize it and utilize it and move it around, but there's typically stress associated with that. So when we developed our most recent interface, and it's still ongoing, there's some new tools coming, but the mobile and immobile indicators they're all set up on the detailed comparison report. So if you go to the detailed comparison report and you scroll all the way down to the bottom, you'll see there's immobile indicators and mobile indicators. And they're actually telling you which one of those nutrients is high and which one is low on that that very, very small chart at the bottom. Very, very powerful chart. Extremely easy to sum up the report. When I first started getting into writing a lot of recommendations from our reports a bunch of years ago, we used to religiously look at that. Since then, I utilized a slightly different technique. But there's so much information in those immobile and mobile indicators that on the detailed comparison report that I'd really encourage you to go back and look at a report or look at one of the sample reports online to actually go drill down, because it's just what we're talking about here. It's showing you where those deficiencies are showing up. So target range charts and just your overall low, medium, high on the basic report, those are very good indicators of telling you, what's actually happening in the plant. Unfortunately, what we've realized with plant sap analysis, and this is where we diverge from every other lab out there, is that it's not all about the levels, right? There's a lot more going on within the plant than just pegging a sap analysis to a 1800 part per million calcium. We know that's going to be the key. Not necessarily. We've seen plants in both directions where plants maybe have, where we would think our target range chart would be, 1800 to 2000 part per million calcium. And then we've seen plants that were sick that had, 4000 parts per million calcium. And conversely, we've seen plants that are healthy that had 800 parts per million calcium, on the same crop, in the same cultivar of the same, basic crop. So those target range charts are set up to give you a basic guidance, but the deeper layers of understanding sap testing, from our perspective, really have to go through the detailed comparison report, because there's a lot that's in the balance of new leaf to old leaf that can be drawn and teased out from there. There's also, on the detailed comparison report, there's ratios for salinity and turgor pressure. So your calcium to potassium ratio for turgor pressure and your calcium to sodium ratio for salinity. Those are very potent ratios. And when you start to get into, oh yeah, we've got huge amounts of sodium, it's going to correspond with a sodium stress and probably a visible plant problem. So I hear a lot on the airwaves now about everybody wants to decrease input. Well, how the heck are you going to do it? Right? Do you have a strategy? Do you have an approach? Do you have data points? Well, we do. We've done it over and over for hundreds of customers. And so if you really are serious about saving money on fertilizer, I would really encourage you to be serious about some of the leaf analysis, because that's how it's done. What we found, and we'll learn about this next week, it's the very first principle of nature is that plants are able to survive on low levels of nutrition delivered by microbiology. Carbon is king. Thanks again very much, and look forward to seeing you all next week. Take care.